We will now discuss the formation of peptides, which is the linking of the amino acids from the previous section, along with the first couple parts of the next section that talks about how these are all related and how they build upon each other to make the proteins. A peptide are the amino acids that are then linked together to form a long chain. It's not necessarily a polymer because it's not the same thing that's repeating over and over and over again. However, there are the 20 that are placed and intermingled into a line. So they are composed of small parts that are linked together to make a really long chain, but it's just not a repeating chain. So a peptide bond, if we look at the amino acids, you'll notice that there's an amino on one side and a carboxylic acid on the other. And that is true through the whole thing. So notice that we can link these up using the exact same type of bond because it's the exact same compounds, exact same functional groups. Carboxylic acid can link to amine. Carboxylic acid can link to an amine, so on and so forth. So when we're looking at peptides, a peptide bond is the linking of these amino acids. If we look closely at the carboxylic acid and the amine, and then compare it to the peptide bond itself, notice that this carbon is linked to a double bonded oxygen as well as an oxygen to the right on the top. When we take it down to the bottom one, there's only one oxygen there. It lost an oxygen in that process. On the right hand side, if we look at the amine, it has 3 hydrogens here. However, if we look and compare it to the nitrogen down here, it only has 1 hydrogen. It lost 2 of its hydrogens. So we've lost 2 hydrogens and 1 oxygen. So in the process of making this bond, of linking the carbon and nitrogen together, it's a dehydration reaction. We've taken out water and linked the two together. So for instance here, this oxygen and 2 of these hydrogens would go away. What would be remaining is the carbon-oxygen double bond, then is bonded to the nitrogen, and that's also bonded to a hydrogen. The rest of it is going to be exactly identical to what's up here. So notice, here's the SER, here's the SER. It didn't do anything different. THR, look here, it's the THR. It's exactly the same. The only thing difference is water is taken out and there's a carbon-nitrogen bond there because it's now linked together. So a peptide bond is the linkage of these amino acids. Now peptides are long strings of these amino acids linked together. To name them, they essentially take the original name of the amino acid and replace it with a YL. So notice alanine becomes alanyl, glycine becomes glycyl, so on and so forth. The last one doesn't change. So then you put it all together into one word. But more likely you're going to see it with the abbreviations of the individual amino acids. Now the beginning and end of the chain, we have the N-terminal and the C-terminal. The terminal just means end. So the N end and the carbon end. So N stands for nitrogen because nitrogen's at the end. C stands for carbon because carbon's at the end. And that's how they determine it. Since it always goes nitrogen, carbon, nitrogen, carbon, nitrogen, carbon, you're going to have a nitrogen finishing up on one end and a carbon finishing up at the other end. And that's just identifying which is which. Now we're going to discuss the first 2 levels of the protein structure. The 3rd and 4th level will be discussed in the next section. So the primary structure of the protein are the peptide bonds. The peptides themselves. The linking of the amino acids. It's telling you the actual sequence of those amino acids. So notice it says that ALA is first, then LEU is second, so on and so forth. That's what the primary structure is. They call it the backbone because it's the original. It's what everything's based upon. Then we get to the secondary structure. Now we're talking about how the structure interacts with other structures or even with itself. For instance, the alpha helix is actually interacting with itself. Think of a line and then it twists upon itself. It's the same line, except it's curling down like a telephone cord. This is part of the carboxylic acid there. It's the carbon-oxygen double bond from the carboxylic acid. This nitrogen-hydrogen here is from the amine. Notice that it's actually being shared from top to bottom. It's not left to right, but it's getting its spiral structure from the attraction of the oxygen, the partially negative oxygen, to the partially positive hydrogen from above and below it. That is called, once again, the hydrogen bond. A hydrogen bond is not a true bond. It's an attraction due to electronegativities and polarity. Another secondary structure is the peptide sheet, the beta-pleated sheets. Contrary to the alpha helix, where it's bonding to itself, the beta sheet is actually bonding to multiple sheets at the same time. With the alpha, the R bonds, the different amino acids that are chained to these carboxylic acids and amines, are actually coming out of the circle, out of the spiral. With the beta sheets, they're coming into the computer and out of the computer. Side by side, notice that it's still going to have that hydrogen bond. It's still going to have that attraction. So here's that carbon-oxygen double bond from the carboxylic acid. And here's the nitrogen-hydrogen from the amine. The oxygen is still getting attracted to the hydrogen, making that hydrogen bond. But instead of being attracted to one within the same peptide, it's actually attracted to the next peptide over, and it's making kind of a sheet. And it's pleated because of the shape of the molecules themselves. So let's go back and make sure we covered everything. Peptide chains side by side, whereas alpha helix is within the same and it curls. Hydrogen bonds between the chains. Both the alpha helix and the pleated both have hydrogen bonding. R groups are above and below the sheets, where with the alpha helix, the R groups are facing outward. And some examples are silk. And finally, the third secondary structure that we're going to discuss is the triple helix. So this is essentially 3 alpha helixes wrapped together. Think of it as like yarn or some other kind of string. So you have, individually, these are fairly weak, but once we get 3 of them wrapped together and coiled around each other, it becomes stronger and stronger and stronger. Notice that it's really strong with collagen, connective tissues. It has to be strong for those purposes. Tendons, cartilage. So it's just alpha helix, but it's 3 of them wrapped together. One example of this is a fibrous protein. Long fiber-like shapes. Keratin, which makes up our hair or wool or skin or nails, is actually made of this triple helix. Whereas feathers are made up of the beta carotenes, which are the pleated sheets. So they'll be flatter. Alright, so let's do a little review. Polypeptide chains side by side. The fact that it says side by side tells us it's going to be the pleated sheets. Sequence of amino acids. So this is the backbone. This is what's telling you what's bonded to which amino acid. That's the primary. Corkscrew shape with hydrogen bonds. The fact that it's a corkscrew shape tells you it's either alpha or triple helix. Well, if we look at the next one, it's 3, so that's going to be a triple, so that means this is alpha helix, where the 3 is triple helix. We're going to stop there, and we're going to talk about tertiary and quaternary in the next video.